So I'm going to first introduce the first session. We're really excited to have speakers from other labs come and tell us about how they're developing and using Miniscopes. And so Susie Feng from my lab and Zach Pennington, a postdoc in Denise Cai's lab, are going to moderate this session. So I'm going to throw it over to them.
All right, thanks so much, Tristan. Hi, everyone. Again, I'm Zach Pennington. I'm a postdoc in Denise Cai's lab, and I'm joined by Susie Feng, graduate student in Tristan Shuman's lab here at the Icahn School of Medicine at Mount Sinai. And we're both really thrilled to get to moderate this series of exciting talks designed to highlight some of the most cutting-edge Miniscope research being done, novel developments in both hardware, software, and analysis, and also highlighting what you can gain from using these tools in your research and the sorts of insights that these tools provide. I want to take a second to just thank all of our speakers for coming and sharing their time with us. We know they're really busy and just lead really productive labs. So we'll go ahead and get started in just a second. I'm going to say we have each speaker will go for about 15 minutes, and then we'll have 5 or so minutes for Q&A afterwards. So please go ahead and use the chat or raise your hand and during those periods, and we'll try and get to as many questions as we can. And if not, we're going to have the breakout rooms afterwards. So please join those and ask speakers more questions there. So we'll go ahead and get started with our first speaker, Dr. Caleb Kemere, Associate Professor of Electrical and Computer Engineering at Rice University. Caleb is a huge contributor to the open source community, serving on the board of directors for the Open EFIS project, and his lab's research focuses on designing systems to interact with complex neural circuits. These developments are being pursued in his lab in the service of closed loop control of hippocampal circuits, optimizing deep brain stimulation techniques, and other neural interface technologies. Today, we're really excited to hear about recent developments in his lab focusing on faster and more sensitive imaging methods. So with that, I will hand it over to Caleb.
Awesome. Thank you for that introduction. I should point out, so the Open EFIS company team is sort of evolving, and if we had an in-person Society for Neuroscience, we might have disbanded the board of advisors. So I may no longer be part of that, because I think now they're converting to a nonprofit company. I love the Open EFIS team. I feel like I can't speak for the Miniscope folks, but I know that they were an inspiration to me in terms of wanting to develop and build tools that would be shared by the community and really enable stuff. And so it's so cool that, like, at least in terms of the store that you guys are partnering with them. I'm going to share my screen, and hopefully this will work. Are you seeing my slides? Awesome.
Yeah, you're good.
Great. And you can hear me okay. Awesome. So today, what I want to talk about, because I think it's probably the most interesting thing to talk about, is work that was done in large part by a really excellent graduate student in my lab, Jill Junot, who now sadly is no longer in my lab. She decided that she wanted a real job and to be in charge of an engineering team and to do cool stuff. But she's still sort of working a little bit on the side on things, and I'm going to talk at the end about where we're going with this. The thing that I want to present to you today is a variant of the Miniscope V3 that Jill developed starting about 3 years ago. There's a paper on bioRxiv, so if you want more details, then I'd refer you to that. And we hope that paper will be done soon, and I'll talk about that a little bit at the end. The main thing that the MiniFAST differs from the original Miniscope is in that we used a different image sensor. And I'm going to talk a lot about why the image sensor that we chose is potentially more useful in certain cases. But I'll point out that there's one other little tidbit that we did, which was that we developed a 3D printed connector for the base and base plate. We use this for a lot of VR experiments, and we find that it's really nice to be able to put on the Miniscope and just sort of twist it on, and there's this magnet that locks it into place. This was actually developed by a freshman at Rice. So those of you who have children who are aspiring engineers, this is just Rice is an awesome place, and we have really good students. I think if you're trying to do chronic freely moving experiments where you want to see the same field of view across days, it may not be quite as rigid as the new V4 mounting system, and so I don't fully endorse this for that. But we have actually also adapted, so we have a version of this for the V4 as well. Please reach out if you're interested in that. All right, so that's all I'm going to say about that connector, but feel free to ask questions at the end. All right, so when we started working on this project, we had this PCB that Daniel Aharoni had built, which was using an image sensor from ON Semiconductor. And I should just say, when I saw this, I was like astounded. I thought, how is some random physics postdoc able to do such amazing engineering? And then he sort of revealed the secret, which was that there's these special chips that allow you to have this connection between an image sensor and a USB connector that make it slightly less impressive, but still, it's really a wonderful work of art. The original one, and then now the V4 is even more impressive. But what's interesting about the original sensor is that the resolution is actually fairly low, and the maximum frame rate is also somewhat low. So we looked around and identified at that time what the sort of the best image sensor in this size range on the market was, and that was the family of sensors made by Sony that they call them Starvis. They're backside illuminated CMOS, so they're sort of at the cutting edge of technology in terms of making an image sensor that's maximally gathering photons. And in the game of fluorescence imaging, maximally gathering photons is sort of the most important thing. So the things that I'm going to talk about as why maybe this sensor is nice, the things that it gives us versus the V3, and I'll talk a little bit about the V4 along the way, is that it has a much larger resolution, so full HD resolution, so 2 megapixels instead of half a megapixel, and it is more sensitive to light, even though the pixels are smaller, and it can image much faster. Okay, so this is in terms of size, this is me attempting to scale the V4 image sensor area and chip. So the V4 is actually the image sensor used in the V4 is a little bit smaller than the old one, but it's actually pretty comparable. So this is a comparison of all of the Miniscopes that we could find in the literature in terms of what image sensor they were using. This is showing the pixel size, the resolution, and then also the maximum frame rate. So the V4 lies here. It's also a half megapixel sensor with 4.8 micron pixels that can go up to 120 frames per second. So I'm going to show you some data today where we've imaged at 500 frames per second. All right. So why is a different sensor a potential advantage? All right. So first of all, resolution is complicated. And I'm an electrical engineer, but I don't do imaging is not my main bread and butter. And I recognize that there's people who understand this sort of thing really, really well. And they're probably on this panel. And so I'm not going to try to pretend to explain to them. My simplified understanding is sometimes the optics constrain your resolution. So no matter what image sensor you have, you're going to see something that's sort of blurry. But if your optics are good enough, so if the size of your blur is small relative to the pixel size, if you have more pixels, you get more resolution. And there's a couple of benefits of more resolution. So one is potentially, now we haven't seen this, and I'm sad I'm missing the breakout room on data analysis, because the fact is that we often find that when we have larger resolution, it just makes our data analysis harder because there's just more data. But in principle, it seems to me as a signal processing person that more data should be better, and so increased resolution should give us better reconstructions. We certainly see this with our computational cameras. I'll show you that in a second. But then also you can scale, so you can image over a larger area by using less magnification and see a larger field of view if you have increased resolution. So here is an example of this is the classic, for those of you who don't do optics, there's this slide that you can buy that the US Air Force figured out 70 years ago that shows different sized line pairs. And it's used to calibrate image sensors. And so you can see here is a comparison of the V3 and our MiniFAST sensor imaging group 9. And you can see that the image quality, so the ability to resolve these line pairs, is indeed better. So increased resolution does give us, so we do actually see increased resolution with the optics in the Miniscope. All right, so now in terms of field of view, I'm not going to show you any data, but I'm going to show you a tweet from Suhasa Kodandaramaya at the University of Minnesota. And those of you who aren't on Twitter might not have noticed this. Those of you who are might have noticed it, but you probably didn't zoom in at the picture. So his lab and then also a lab at BU have both been trying to develop versions of the Miniscope that could image larger chunks of the brain. And we're actually collaborating with both of them so that they can use the MiniFAST image sensor board for their system. And if you had zoomed in on his tweet, you would have noticed, oh, this is strange. There's actually 2 different kinds of Miniscope that he's shipping out to these groups that want to try his system. One is using the traditional V3 Miniscope sensor, and one is actually using our MiniFAST sensor here. So this is the little zoom. But just also a shout out to the Davison lab at BU, who's doing something different but the same. All right, so I want to show you a video here, and since who knows how well this is going to go over Zoom. Let's see, move that out of the way. So this is a comparison using the same GRIN lens of a V3 imaging a field of view and the MiniFAST imaging a field of view. And I always like to just show these videos. And those of you who do the reason you're here is because you're interested in calcium imaging. It's just every time I see these things, it just makes, I just sort of want to stop and admire what the brain looks like, right? We actually are seeing neurons. The neurons are actually being active. This is super cool. But this is showing the increase in resolution and how we potentially can resolve things like dendrites better with increased resolution, right? So presumably that would be the application of higher resolution in this particular context. All right, let me find my slides again. So I should have mentioned my funders. So we have the NIH and DARPA have both been helping us to develop the system. And the reason that they've been doing that is because they're excited not about Miniscope, sadly, they should be, but instead because they're excited about new devices that we've been building with collaborators. So the collaborators on this project are Ashok Viraghavan and Jacob Robinson, where instead of using lenses, we use very carefully designed phase or amplitude mask right in front of the image sensor. So here's the image sensor with a little spacer. Here's this phase mask. And then these are filters. And the idea is that what you see when you take a picture with one of these devices is something very blurry. But then when you pass it through a reconstruction system, you can recover the thing that the scene that you've looked at because of the way that the image has been, the scene has been processed by this phase mask. Laura is here. She's going to talk about this. So I'm not going to talk very much more about it, except to say that what we found is that having more pixels improves the reconstruction quality in these systems. All right. So increased resolution is nice. It gives us potentially the ability to do better computational imaging. It also gives us the ability to look over a larger field of view or potentially see things like dendrites. So what else is good about our new image sensor? Well, it's backside illuminated CMOS, where the actual number of electrons that we get per photon is improved, is significantly improved. Why is that useful? So why is increased sensitivity per se valuable? Well, one reason why it's useful is because one of the things that limits our ability to image over long periods of time, this very much depends on the protein that we're imaging, is photobleaching. So photobleaching is this surprisingly mysterious phenomenon that, again, there's much more expert than me people on the panel about this topic. But my understanding is basically that you have these fluorophores that when you hit them with light, they go into this excited state, and then when they relax back down, they emit a photon, and that process of vibrating from excited to unexcited state eventually breaks them. And you have millions of these proteins expressed in your neurons and constantly being generated by the viral or genetic manipulation that you've done to the animal. And so you're constantly adding new fluorophores, and you're constantly losing some to this damage or just to the cell eating them. But if you excite them with a lot of light, then they do this bouncing a lot, and they tend to break faster. And so you see this phenomenon where, with a high intensity of light over time, the thing that you're looking at becomes more dim. And if you use less excitation light, you're exciting fewer of these molecules, fewer of them are breaking, and you see something that doesn't seem to dim as quickly. Now, there's some complexity here, because our ability to do the automated matrix factorization, reconstruction of where the neurons are, or even things like image stabilization, probably depend on getting a high quality picture. But in general, if we can use less excitation light, then we will bleach less and it will be better. Now, when I say if we can use less excitation light, what I mean is if we can still see the scene, then that will in general be good. And so having a more sensitive image sensor allows us to still be able to see the fluorescence from the neurons that we're interested in, even when the excitation light is minuscule. So there's some benefit of reduced photobleaching. And then the dream is that what this enables is it enables us to image a much longer period of time. Now, these pictures are taken from a transgenic Thy1 GCamp6F, like the classic Thy1 GCamp mouse, which photobleaches a little bit faster than AAV-based GCamps. Then also, of course, the GCamps are constantly being developed. The newer ones are brighter, less sensitive to photobleaching. But in general, there will always be a new fluorophore that wants to photobleach that we don't want to photobleach. And so this is something that I think is important. This is just a random aside. I don't know how many of you have tried to image the Thy1 GCamp6F mice, but their GCamp is really fast, surprisingly fast. The transients have like really spiky peaks. And we noticed this and we're like, whoa, this is crazy. So this is showing the second reason for sensitivity, which is that when you image fast, you have very few photons that come per frame. And so increased sensitivity allows you to do a better job of reconstructing these. Of course, people that typically are imaging at frame rates of hundreds of hertz are using very expensive cameras that are cooled and have very, very low noise. And so the question that we are really still asking is whether, in addition to nice fast calcium transients like we see in the Thy1 mice, if we can see genetically encoded voltage indicators. And so this is using the sensor over a Petri dish, a microscope slide that has cultured hex cells on it that are being driven to spike, being depolarized while expressing a genetically encoded voltage indicator. And we can, in fact, resolve these spikes. We image at 500 frames per second. And this is a picture from our collaboration that we've recently established with the Pierrebonne Lab. They've been working with us on our DARPA program. They are developing some really cool new voltage indicators, including red-shifted red and green positive going voltage indicators, as well as sort of classic negative going indicators. And this is a picture from their preparation. This is a window over layer 1 neurons. This is a layer 1 a mouse expressing. This is, again, ASIM neon. And we can see that at 500 frames per second, we're definitely seeing the neurons. But at the moment, we're not resolving spikes. It seems like our signal to noise level is not quite high enough. And so we're trying to boost our excitation light even a little bit more beyond what the normal LED on the Miniscope provides. But I think we're very close to being able to image GEVIs. All right. So the last reason why speed is nice. So I should mention something. So the Sony Starvis sensors are rolling shutter, which in general is not ideal. But one of the things that allows is that allows these very, very high frame rates. When I described the sensor at the very beginning, I said that it images full HD. We can't actually image HD at 500 hertz. HD at 500 hertz is a very, very high bandwidth. And what we can do is we can image 500 hertz through a stripe of the image sensor that we can select. So it allows us to select a subset of the pixels and image them to allocate these pixels per second to a smaller range of the sensor. And so the applications here for fast imaging are ones probably where you would take a snapshot of the full field of view and then you would select some subset of it to image faster. But I'm going to talk about that again at the end. Okay. So why else might we want to be fast? We want to do closed loop interfaces. Then we care about latency. If we're imaging classic calcium frame rates of 15 frames per second or 30 frames per second, our latency is limited by the frame rate. But if we're imaging at 500 hertz, all of a sudden our latency can be driven much more by the actual physical interface to the computer. For example, the USB interface, or potentially an FPGA version of the interface. So this is a note to myself to shout out to the Blair Lab at UCLA that I know has been building a closed loop system that has an FPGA that's actually doing some real-time processing on board. But these limits, you only actually get to these limits if you've gotten past your frame rate limit. And so I think high speed imaging is something that I think will enable really cool closed loop stuff. With electrodes, we already are doing closed loop, really cool experiments. We would like to do those with optical interfaces as well. All right, so where are we going with the MiniFAST? So our dream is really to show that we can use this for in vivo imaging of GEVIs, and we're hoping that we'll be able to do that here in the next few months. It's been actually, I think we have a very kind editor at the journal to which we submitted our paper, who asked us to do this and has given us now many months, and we're still trying to find exactly the right combination of sensor and mouse model that will allow us to have a stable expression that we can use to test this with. We're hoping to integrate this with the V4 platform, but our number one priority is actually to change the way that the sensor integrates with the system. So this is just a little bit of geeking for the electrical engineers who are listening. So most of the Miniscopes that people are using this really nice chipsets. Yes.
I hate to interrupt.
Am I going long? Yes.
Going a little bit long, and we have to get to the.
Sorry, I'm done. I'm done. But the last point is we're limited by everything else in the pandemic. So sorry. Thank you for interrupting me. I wasn't sure what time we started.
No, that's fine. I'm sorry to interrupt you. Thank you so much for coming. This is super exciting.
Yes.
For anyone who has questions, please feel free to post them in the chat. And then also we're going to have the breakout rooms afterwards where you can talk to Caleb more about this if you're interested. And we'll now move on to the next speaker.